All right. So we deal with a lot of carboxylic acids in metabolism, but actually at the pH in our bodies where this is happening, the pH is slightly greater than 7, um, so it's slightly basic, and so carboxylic acids are dissociated. So in our bodies, carboxylic acids are actually in their carboxylate ion forms when they take part in reactions, like the citric acid cycle. So for example, in water, succinic acid will be in equilibrium with its carboxylate ion because of the ionization due to being in water. Um, Carboxylates are part of the metabolic processes. In glycolysis, for example, a molecule of glucose is broken down. Uh, Glucose gets broken down into two molecules of pyruvate, which is the carboxylate salt of pyruvic acid. And then during strenuous activity, when oxygen levels are low, so anaerobic, so that's why reduction happens instead of oxidation, pyruvate can actually be reduced to give lactic acid or the lactate ion. We'll go over some of these processes when we get to the metabolism part of this semester. In the citric acid cycle, formerly known as the Krebs cycle, di and tricarboxylates are oxidized and decarboxylated, so they lose carbon dioxide, decarboxylated, to produce energy. Citrate with 6 carbons is oxidized to a 5 carbon alpha ketoglutarate. And as we breathe out CO2 and we make energy, we can continue to live. Um, I'll write that down. Decarboxylation. We'll talk about that later, again, when we get to later chapters. The citric acid cycle continues as alpha ketoglutarate loses a CO2 to give a 4 carbon succinate. So this was the molecule we started talking about. And just here, losing one CO2 brings us down to 5 carbons. Losing another gets us to succinate. Succinate then, in a series of reactions, will get converted into oxaloacetate, and the citric acid cycle is ready to accept another molecule of glucose. And we'll go through that cycle, but you can see here lots of different carboxylate ions are involved in these metabolic processes. And that's because they're more soluble in water, in our aqueous water environment. Let's talk about esters. By the 1800s, chemists had discovered that salicin, excuse me, I have to pull up my notes, I'm sorry. All right, salicin. Salicin from willow tree bark, so that's a willow tree. Um, Salicin from the willow tree bark and from the leaves, if you put it in water and you drink it, could actually relieve pain. So what was happening was that the body was converting salicin into salicylic acid, which can irritate the stomach lining, but we would get some pain relief from this compound. Now Bayer, the manufacturer, discovered that an ester form of this compound, called acetyl salicylic acid, was less irritating to the stomach than the salicylic acid, and was still effective. So we're going to look at some esters and some how we make esters, but you'll see that there are ester compounds that we can use as pain relief, and so they do some good for us. And we'll learn why The salicylic acid was also bad for the stomach, why it irritated the stomach lining. We'll see that structure. But the learning goal here is to be able to write IUPAC and common names for esters and to be able to draw their condensed and line angle structures. So let's take a look. Esters are synthesized from the reaction of carboxylic acids and alcohols. These esters are found in fats and oils, and they're responsible for many of the aromas and flavors from things like bananas and oranges and strawberries, pears and pineapples. So you can see some ester here. So here we have, actually this isn't an ester, this is just a carboxylic acid. And here we have that same carboxylic acid as an ester. So what happens is we bring in, let's say, an alcohol like methanol. We're going to get whatever's attached to our alcohol getting added to whatever was, or I guess in place of, the hydrogen on our acid. So we get this situation. So an ester has our carboxylate with an alcohol left over, kind of mixed together. The way that we name these, I guess we'll go over that in a minute, but you can kind of see it here. We're going to call the part that's left over on the alcohol by a substituent name, in this case methyl from the methanol, and then the leftover of the acid is our carboxylate. So in this case it was acetic acid, so acetate. It could also be ethanoate, methyl ethanoate. Esterification is what we call the reaction between the carboxylic acid and alcohol. We usually need an acid catalyst and some heat to make this happen. The hydrogen gets replaced with the alkyl group from the alcohol. Here it's color coded so you can see alkyl group on the alcohol. It's going to replace this H. Now, an equilibrium reaction means that this is subject to forming an equilibrium. We may not get all of our acid converted into the ester. And so there's a couple of ways that we make this shift this direction. We can either add an excess of reactants, so just have lots of this stuff, or an excess of one of these, whatever one's cheaper, will drive the reaction to the right. We could also remove products as they form. So if you have some way of pulling the water out of this as it is forming, that'll keep the reaction moving to the right. That's part of the process that we use to make esters. Quick learning check here. Draw the condensed structural formula for the product of propionic acid and methanol. So in this case, you're making the ester. So remember, take what's attached to the alcohol and attach it to the carboxylate. Let's go ahead and pause it, give it a shot. You should be taking this CH3 and sticking it on this O. Technically, this O right here, part of the alcohol, is this O, but we're not going over mechanisms, so we don't really need to see how they're moving around, but if it helps, this part right here was the alcohol O. There's also water that forms, don't forget that. When we have the OH and the H from our alcohol pairing up, they make that water. So the Bayer company, as I mentioned, was able to take the salicylic acid and actually make a ester out of it. So if you can see salicylic acid here, whenever we ingest this, we get the salicylate ion. Sorry, what is it? Salicin from the oak tree becomes salicylic acid in our bodies. This has not one, but two acidic parts. This is why, well, I'll tell you why old aspirin smells like vinegar in a second, but this is why this molecule was so harmful to the stomach lining. It's because not only does this H come off, but this phenol group here is also acidic, and so that H would also come off. Now, you could react this molecule with acetic acid to produce an ester, and so the ester that was formed in the acetylsalicylic acid, or the acetylsalicylic acid, or aspirin, was an ester bond formed here on the phenol. Okay, but it still has the acidity here up at the top. So this was slightly less harmful to the stomach, but just as effective because it still has, I guess, the important part of the molecule. We haven't talked about that stuff yet, but this shape is what is letting your body get some pain relief. So long as we don't mess with that shape, it still works. Now, because this is an equilibrium reaction, that means if you have an excess of water or aspirin, you can actually get the backwards reaction happening. So sometimes old bottles of aspirin start to smell a little bit like vinegar because over time, water from the air will react with the molecule, hydrolyze that ester bond, and give you back acetic acid, which is what we think of when we smell vinegar. Now, this is oil of wintergreen. You can see methyl salicylate. Now this has a pungent minty odor and flavor. It's used in skin ointments. You can see it's from the same starting compound as our salicylic acid and aspirin. This one, if we mix it with methyl alcohol instead of what I guess was acetic acid, and actually, oh yeah, so sorry, just to be clear here, the ester is forming not between salicylic acid and an alcohol, but between acetic acid and the phenol. So this is the alcohol that's reacting, and this is the acid that's reacting. In this case, salicylic acid is the acid, and we're reacting that with an alcohol. So it's going to be an ester bond is going to form here. So it's important that you know where your bond is going to form. Okay? The reason this bond formed down here is because this was the alcohol. And whatever was attached to this O would just get attached to the carboxylate ion of acetic acid. And that's what happened. Over here, we're reacting an alcohol with the salicylic acid. Now, the ester bond is going to happen right here, and whatever is attached to the O on this side is going to attach to our salicylic acid, and you can see that here. So we still have the phenol group, but now we have this ester bond. This is used in skin ointments. It can pass through the skin. It acts as a counter irritant. It produces heat to soothe the sore muscles. It's a really good compound, but has different effects than aspirin. Here's one more. This is Dacron. Dacron has many uses. It's a permanent press fabric. It's used to make artificial blood vessels and valves. It's made into a film called mylar. Maybe you've heard of mylar. And a plastic known as PETE, or polyethylene terephthalate. And it's used to make plastic drink bottles. You can see the ester here is formed between, so this is a polymer. So this is a long repeating sort of, I guess, chain of these two monomers. One of the monomers is the acid, the other monomer, so this is a dicarboxylic acid, this is a dialcohol. So you can get a link here, alcohol and carboxylic acid, and then this side of the next molecule will link here to this side, and then we just get a repeating chain. So sets of these two guys, this guy and the alcohol, and then this guy and the alcohol, and over and over and over. Those are ester bonds, polyester dacron. So lots of uses for ester bonds, not to mention the natural aromas. Let's talk about those. Esters such as ethyl butanoate, I mentioned, can provide flavors of fruits. This one is actually pineapple. So how do we know this is ethyl butanoate? 1, 2, 3, 4. So this is butanoic acid. So butanoate. And then ethyl, because this would have been ethanol if it was the alcohol. So ethyl butanoate. We always name the alkyl part first, then the parent chain of the alkane, of the carboxylic acid. This is kind of a way to show it to you. So the first word indicates the alkyl part from the alcohol. When you look at your when you look at your esters, you need to be able to tell which side came from the alcohol and which side came from the carboxylate. The second word in the name is the carboxylate ion, named from the acid. So you can see here in this compound we've got methyl from methanol, and we've got this would be ethanoate from ethanoic acid. So you could call this methyl ethanoate, or if you use the common name acetic acid, this would be acetate, so methyl acetate. There are common names for these, don't forget, and we do use them quite often. Esters also have IUPAC and common names. So the IUPAC name is what we just went over. The common name is a little easier. Let's talk about those. So you can see here, so ethyl acetate, I don't know that that's a common name, that just sounds normal. Oh, I guess, sorry. So common names being that we can use some of the old naming. So remember with our carboxylic acids, we could say formic, acetic, propionic, butyric. That still works here. So while this is ethyl acetate, it can also be ethyl ethanoate if we're being more specific, IUPAC. This could be common name methyl propionate, but it could also be propanoate if we're using IUPAC. What just happened here? I think my pen is threatening to stop working. Ethyl benzoate here, benzoic acid being benzoate. There's no common name for this one. So just some examples. And if you wanted to do, if we were doing substituents, like if this had a, yeah, but I think my pen died. Fantastic timing. So if we had a CH3 on there, this would be methyl 2 propanoate, or alpha methyl propanoate. So remember, substituents can also be added in here. Alright, naming check. So go ahead and try to guess the name of this, the IUPAC name of this ester. Or I guess not guess it, but try to name it. So pause it, give it a shot. So hopefully you came up with something like ethyl propionate or ethyl propanoate. Got another one. Give the IUPAC and common name for the following compound. This one is responsible for the flavor and odor of pears. So here, hopefully you came up with the propyl part is the remainder of the alcohol, propyl. This is 2 carbons long, so this is either ethanoic acid or acetic acid, so ethanoate or acetate. No substituents here, so we don't have to worry about naming anything with numbering. Alright, I've got less than 20 slides. I guess I'm going to try to push through this. I'm sure my kids are going to wake up here any second. So, many of the fragrances of perfumes and flowers and flavors of fruits are due to esters. Small esters are volatile. Volatile. Meaning that they we can smell them. They evaporate really easily into the air. They're also soluble in water, so we can taste them. For example, the odor of grapes is due to an ester. I'm not sure odor is the right word, but here's a couple of those compounds. So you can see here, propyl ethanoate is what gives pears that flavor. Pentyl ethanoate is bananas. I've also made a compound. We do this in organic chemistry. We make something called isopentyl ethanoate. Isopentyl. It's our ester chapter. This stuff also smells like bananas, surprisingly. Oranges, pineapples, apricots, you can see here. There's And this is not an inclusive list. There's a lot more. So a couple of learning checks here. Draw the condensed structural formula for each of these following compounds. So this is naming, taking the name and drawing the structure as opposed to drawing the structure from the name. So try to draw propyl pentanoate and ethyl propionate. So I'm mixing some common name and IUPAC name here, but give it a shot. So propyl came from, hold on, did I draw? Yeah, propyl, 3 carbon. Sorry, I was thinking pentyl, I got them mixed up in my mind. Propyl is 3 carbon, so as long as you see 3 carbons on the alcohol side, and then pentanoate is 5 carbons. Ethyl, 2 carbons. Propionate is 3 carbons. So this takes a little bit of practice. Now, if we can make esters, we can hydrolyze them, so I'm sure that's next. Let's talk about properties of esters. Now, ethyl acetate, as I'm showing here, is the solvent in fingernail polish, plastics, lacquers, that sort of stuff. The goal here is to describe the boiling points and solubility of esters, to draw their condensed and line angle structures, and to draw the products from the acid and base hydrolysis. So breaking them apart. So first, esters have higher boiling points than alkanes and ethers. Remember, ethers do have oxygens, alkanes don't. But the boiling points are lower than those of alcohols and carboxylic acids of similar mass. So esters don't have the same hydrogen bonding ability, so they're not as high boiling point as the alcohols and the carboxylic acids. And again, it's because they don't have those hydroxyl groups, so they can't hydrogen bond to each other. They can hydrogen bond to water, which makes them very soluble, but in terms of hydrogen bonding to each other, you can see here, with my terrible drawing, there's no H here anymore. There's no H on an O, so now all this can do is just be polar. There's no H's to grab anywhere, so decreased boiling point. You can see here, though, with water, definitely still soluble. Esters are soluble if they have at least under 5 carbons. As the number of carbons increases, of course, their solubility decreases. The partial negative on that oxygen is what will allow the hydrogen bonding with the water molecules. So esters are soluble in water. Let's talk about hydrolysis. Hydrolysis is cutting with water. Cutting with water with an ester is always going to give us back our alcohol. So we always get back our alcohol. Now, we don't always get back an acid. You might be saying, but wait, to make an ester, we use an acid and an alcohol. You're right. But depending on the pH of the solution, or in other words, depending on what we use to break this ester, we may not be left with an acid. For example, if I use water to break this apart, as I'm showing you here, this is water with acid present. Let me circle that. There's acid catalyst here. So the water, of course, goes back in and adds back the H and the OH. So the OH gets added back to our acid. The H gets added back to the end of our alcohol. Okay, now we're good. Because this is an acid, then there's no reason for this OH over here to lose its H, like I told you happens in solutions above pH of 7, like in our bodies. So in acid hydrolysis, we always get back our acid, because acids do fine in acid, and we get back our alcohol. But that won't be the case in base. In base, our acid will not survive. So I'll show you that. Aspirin, stored for too long. We talked about this. This breaks down. It gives back the alcohol. It gives back the carboxylic acid. In this case, we get back acetic acid, and that smells like vinegar. Base hydrolysis. Take a look at it. Base hydrolysis involves using a base, so sodium hydroxide or potassium hydroxide. As such, you're going to be left with two things happening. One, hydrolysis happens, so we get our two parts. But now, instantly, if you were to imagine for a split second, we actually got back our acid. We got back our acid and our alcohol. But now, remember what happens to neutralization? Our acid and our base combine to make water and a salt. So what happens here, we're not going to make water because we're not actually neutralizing the acid here. But what we are doing is we are taking that acid that would have had an H on it, and we're saying in base that H will be gone. And actually, I think a water does form. I don't think I'm showing it here. What am I showing here? No, no water forms. So the OH here, right, doesn't add to our whole molecule. You can see the H adds to the alcohol to give us back our alcohol. And then the O, since there isn't since this isn't a water molecule, there isn't an OH that lands on this guy. So we just get the carboxylate. And anything that's negatively charged should be paired up with a positively charged ion when we finally put it together as a final answer. Let me backtrack again. So in base, we do end up with the two pieces again. We always get our alcohol, but in base, we end up with our carboxylate salt, not our acid. So in acid hydrolysis, we would get back methanoic acid, or in this case, sorry, acetic acid and methanol. But in base, we get back our methanol and we get back acetate ion with sodium. So sodium acetate. So in order to kind of understand base hydrolysis the best, you need to be okay on your neutralization of carboxylic acids, because that's what's happening here. We're just neutralizing that carboxylic acid into its carboxylate salt. And that doesn't happen in acid hydrolysis, just base. There's another situation where we're going to have to think like this. When we get to our amines, we're going to see that we can form amide bonds that can be hydrolyzed. Amides can always give us back either an amine or an ammonium salt, whether it's acid or base. And so it's going to be very similar to this. And that'll come back also when we talk about our amino acids. Okay, I think I'm done. So we have base hydrolysis and acid hydrolysis. Practice those. Ethyl acetate is a solvent used in fingernail polish. Write the balanced equation for the hydrolysis of ethyl acetate with sodium hydroxide. I feel like this is exactly, this was methyl acetate. So now you're going to do ethyl acetate. So pause it, give it a shot. Ethyl acetate, of course, starts off with having an ethyl group on the end. So the only thing different from the previous slide is that we get ethanol instead of methanol. Because this is base hydrolysis, we get the acetate ion, or the carboxylate, and we have to make the carboxylate salt. So we look at who it was paired with. Realize that if there's a situation where we have phenol. Let me go back to that very, very quickly. I think this is the end of the slides here. If we were to hydrolyze this guy with base, realize what would happen. We would get, right? In base, this would be negative. We would get back our, this is the alcohol. We would get back this, and we would get back our acid, which would be the acetic acid. Okay, but in base, the H's would come off. And you're saying, well, why are you taking off the H from the phenol? Well, don't forget, in base, phenol is acidic. So phenol actually has a negative charge. This would have a negative charge, and this would have a negative charge. In base, right, we've got our alcohol back, and we got our acid back, or our carboxylate. If this was sodium hydroxide that we used, then we would have a an Na plus paired here. But realize that this molecule is also very acidic still. Unlike every other example where our ester broke apart into a carboxylate and an alcohol, this alcohol is acidic. This alcohol here not only has a carboxylic acid group on it that would also become a carboxylate, but it also has a phenol, which is acidic, and phenol reacts with base as well. So we would end up with something like this. We would end up with our alcohol actually completely salt form, and our acetate salt form. I like this problem because people often forget that phenol is acidic, and they don't know what to do when there's multiple multiple carboxyl groups in your products. If you add base, every single acid group on here is going to get neutralized. That includes the phenol. This will help you in lab. There's a lab question that comes around like that. All right, last question. Write the balanced chemical equation when methyl acetate reacts with water, heat, and an acid catalyst. So basically this is acid hydrolysis and then base hydrolysis. And methyl acetate is the one that you guys have already looked at a bunch. How about instead of methyl acetate, you try it also with something like, how about, something like that. So that is 2-methyl 2-propanol when it breaks apart. 2-methyl 2-propanol, that's just a tertiary alcohol. And this would be pentanoic acid. Just because, methyl acetate you've done 4 times already. But just to reiterate, in acid, we get an alcohol and an acid. In base, we get an alcohol and a carboxylate salt. Alright, good luck with that, everybody. I will see you in the next video.